Hi, this is Kavita Kundra, and we are now dealing with our next chapter, next section of lipid metabolism. So, we are moving towards section 4. Metabolism of cholesterol. Cholesterol is involved in two major processes, and it is the structural component of cell membrane. Steroid hormones, vitamin D3, and bile salts are derived from the parent compound. Cholesterol is synthesized de novo in liver and intestinal epithelial cell and also derived from dietary lipids. De novo synthesis of cholesterol is regulated by the amount of cholesterol and triglycerides in the dietary lipid. Cholesterol biosynthesis in liver and intestinal epithelium. The biosynthesis of cholesterol, a complex molecule with 27 carbon atoms, start with 2 carbon atoms compound acetyl coenzyme, which is converted to isopentenyl pyrophosphate, and then squalene, some of which are soluble in cytosol and other are bound to endoplasmic reticulum. The process involves acetyl coenzyme gets converted into HMG-CoA, that means 3-hydroxy 3-methyl glutaryl coenzyme. HMG-CoA gets converted into mevalonate. Then mevalonate converted to isoprene based molecule isopentyl pyrophosphate with concomitant loss of carbon dioxide. And IPP gets converted into squalane. Then squalane converted into. In mitochondria, oxidation takes place and acetyl coenzyme is synthesized and is used for cholesterol synthesis. Acetyl coenzyme can also derive from cytoplasmic oxidation of ethanol. All reduction reactions of cholesterol biosynthesis use NADPH as cofactor. Acetyl coenzyme units are converted to mevalonate by the series of reaction that begins with the formation of HMG-CoA. During ketone body synthesis, HMG-CoA synthesized in mitochondria. Here it is synthesized in cytoplasm. The pathway and the necessary enzyme are same as in mitochondria. 2 moles of acetyl coenzyme are condensed in reversal of thiolase reaction, forming acetoacetyl coenzyme. Then acetoacetyl coenzyme and third mole of acetoacetyl coenzyme are converted to HMG-CoA by HMG-CoA synthase. HMG-CoA is converted to mevalonate by HMG-CoA reductase. HMGR, it is present in endoplasmic reticulum. HMGR requires NADPH as cofactor and 2 moles of NADPH are consumed during the conversion of HMG-CoA to mevalonate. The reaction catalyzed by HMGR is rate limiting step of cholesterol biosynthesis and the enzyme is subject to complex regulatory controls. Mevalonate is activated by successive phosphorylation yielding pyrophosphomevalonate. Phosphorylation maintains its solubility. After phosphorylation and ATP dependent decarboxylation yield isopentenyl diphosphate IDP, an activated isoprenoid molecule. Isopentenyl diphosphate is in equilibrium with the isomer dimethylallyl diphosphate DMDP. One molecule of IDP condenses with one molecule of DMDP to generate geranyl diphosphate GDP. GDP condenses with IDP molecule to yield farnesyl diphosphate FDP. Finally, NADPH requiring enzymes Squalene synthase catalyzes the head of the tail condensation of two molecules of FDP yielding squalene. Squalene undergoes two-step cyclization to yield alanosterol. Cyclization is a changing open chain hydrocarbon to closed ring. The first reaction is catalyzed by squalene monooxygenase and squalene epooxidase. This enzyme uses NADPH as cofactor to introduce molecular oxygen as an epoxide at 2 to 3 position of squalene. Through a series of blinded reactions, lanosterol is converted to cholesterol. Then the regulation of cholesterol. The three distinct mechanisms maintain cellular supply of cholesterol. Of these three mechanisms, regulation of HMG-CoA reductase activity is the primary means of controlling the level of cholesterol biosynthesis. HMG-CoA reductase is an intrinsic membrane protein of endoplasmic reticulum. The enzyme activity is extended to cytosol. HMGR is rate limiting enzyme in cholesterol synthesis and is subject to different type of metabolic control. First is feedback inhibition. Cholesterol is a feedback inhibitor of HMG-CoA reductase, thus decreasing further cholesterol. Hormonal regulation. HMG-CoA reductase activity is controlled hormonally by glucagon favors the formation of inactive form of HMG-CoA reductase and hence decreases the rate of cholesterol synthesis. In contrast, insulin favors formation of active form of HMG-CoA reductase and result in an increase in the rate of cholesterol synthesis. Sterol mediated regulation of transcription. The synthesis of cholesterol is also regulated by the amount of cholesterol taken up by the cells during lipoprotein metabolism. Chylomicron remains internalized by liver cells and LDL internalized by cells of liver and peripheral tissues provide cholesterol that causes a decrease in de novo cholesterol synthesis. Chylomicron and LDL are lipoprotein complexes. Inhibition by drugs. Lovastatin and mevastatin are reversible competitive inhibition of HMG-CoA reductase. They are used to decrease plasma cholesterol level in patients with hypercholesterolemia. Degradation of cholesterol. The ring structure of cholesterol cannot be metabolized to carbon dioxide and water in human, and the intact sterol ring is eliminated from the body by conversion of bile acid, which are excreted in feces. The secretion of cholesterol into bile, which transport it to intestine for elimination, is modified by bacteria before lipoprotein metabolism. Lipoprotein are compounds of protein that carry fatty acids and fat-like substances such as cholesterol in the blood. The principal lipid carried by lipoprotein particles are triglycerides and cholesterol, obtained either from the diet or de novo synthesis. Plasma lipoprotein. The plasma lipoprotein are the molecular complexes of lipid and specific protein called apolipoproteins. Lipoproteins are composed of neutral lipid core surrounded by the shell of apolipoprotein. Phospholipids and non-esterified cholesterol are all oriented so that the polar portions are exposed on the surface of lipoprotein. This makes the particle soluble in aqueous medium. Apolipoprotein. The various types of lipoproteins are present along with the different types of lipids and proteins. These are called apolipoproteins. Many enzymes consist of protein molecules along with non-protein molecules. The protein molecules is referred as apoprotein. The lipoprotein are constant state of synthesis, degradation and removal of plasma. Lipoprotein function both to keep the lipids soluble as they transport them in plasma and to provide an efficient mechanism for delivering the lipid content to the tissues. In humans, the delivery system is less perfect. As a result, the human experience a gradual deposition of lipid, cholesterol in the tissue when the lipid deposition causes narrowing of blood vessel condition called LR3. Chylomicron. These are the lipoprotein particles lowest in the density and largest in the size and contain the most lipids, and the smallest percentage of proteins can be seen. Chylomicron functions to deliver dietary triglycerides to adipose tissue and the muscle and dietary cholesterol to the liver. VLDL and LDL are successfully denser, having a high content of protein and lower content of lipid. HDL particles are the densest of plasma lipoprotein. Functions of lipoprotein. What are the functions? It serves as structural component of the particles. It provides recognition sites for cell surface receptors, and it serves as an activator of coenzyme for the enzyme involved in the lipoprotein metabolism. How chylomicrons are metabolized? The chylomicrons are assembled in the intestinal mucosa cell and carry dietary triacylglycerol, cholesterol, and cholesterol esters to peripheral tissues. The particle released by intestinal mucosa cell is called nascent chylomicrons and contains apolipoprotein BHAB48. Chylomicrons leave the intestine via lymphatic system and enter the blood circulation. When it reaches the plasma, the nascent chylomicron is rapidly modified, receiving ApoE and ApoC2 apolipoproteins. And this is your structure. From lymph it goes to the blood. So from that is going towards the liver as well, the blood as well. You can see in this, you can see full mechanism here. And then in the capillaries of adipose tissue and muscles, the fatty acids of the chylomicrons are removed from the triacylglycerols by the action of lipoprotein lipase, which is found on endothelial cell of capillaries. Lipoprotein lipase is extracellular enzyme that hydrolyzes triacylglycerol into monoacylglycerol and two fatty acids. The ApoC2 in the chylomicron activates LPL in presence of phospholipid. The free fatty acid enters the cell passively down a concentration gradient, and glycerol backbone of triacylglycerols is returned via blood to liver and kidney. Patients with deficiency of lipoprotein lipase or ApoC2 shows a dramatic accumulation of triacylglycerol rich lipoprotein is plasma. Example, type 1 hyperlipidemia. As the chylomicron circulates and the triacylglycerol in its core is degraded by the lipoprotein lipase, the particle decrease in size and increase in the density. Since it has lost a considerable amount of lipid component and addition C apoprotein are returned to HDL, the remaining particle left is called remnant. Chylomicron remnant containing primary cholesterol, ApoE and ApoB48 are delivered to, taken up by the liver through the interaction with chylomicron, remnant by hepatic remnant receptor. The recognition process requires ApoE. Chylomicron's remnants bind to these receptors and are taken into the cell by endocytosis. The endocytosed vesicles fuses with lysosomes, and the apolipoprotein, cholesterol esters, and the other components of the remnants are hydrolytically degraded, releasing amino acid, free cholesterol, and fatty acids. And the cholesterol released from the chylomicron regulates the rate of de novo cholesterol synthesis in the liver by causing a decrease in the cell content of HMG-CoA reductase. So, do you find it difficult? I don't think so. This is difficult. This is not complex as carbohydrate metabolism. It's very easy, and I hope you will learn it, because it is actually, if you learn it like a story, you will learn it better. Do it, and all the best for your exams. Stay safe, stay protected. We are covering the last section of lipid metabolism in few days. Thank you so much. Bye bye. Take care.